My name is Sean Hoban. I work at the Morton Arboretum as a tree conservation biologist. I want to start by acknowledging that the Morton Arboretum is located on the ancestral homelands of the Council of the Three Fires, and I acknowledge and respect the traditional caretakers of this land and their ways of knowing. Next slide. My group at the Morton Arboretum strives to produce the knowledge and tools that inform conservation action to improve our assessment of conservation progress and especially to improve our practices ex situ. We mostly use genetic tools, but we also work in other areas such as historical ecology and geography. Next slide. Why do we focus on genetic diversity? Genetic diversity is important for helping small populations survive, including those that might be isolated in urban areas. It's important for helping species survive changes and adapt to new conditions. We know trees are facing climate change and trees are also facing new pests. Shown here is whitebark pine, white ash, and dogwood. And these are some of many trees that are facing pests and disease problems, and genetic diversity can sometimes help these species survive these challenges. And genetic diversity is also protected under legislation such as the Endangered Species Act. Next slide. One of the main challenges with protecting genetic diversity is species geographic ranges are large and we have limited time, money, space, resources, personnel, etc. So we want to do our work efficiently. Next slide. The other challenge is that trees present a diversity of life history, geographic ranges, size, longevity, etc. And we may need to tailor our conservation efforts to different species traits. Next slide. So I'm going to talk briefly about two topics. How do we measure how much genetic diversity is currently conserved ex situ, such as in gardens, seed banks, or urban plantings? And how do we choose what species to focus on for future plantings, such as those species that might need more protection? Next slide. To assess genetic diversity conservation ex situ, we simply sample trees from wild populations, and we sample trees from the ex situ collection, whether that be an urban planting or a botanic garden. Mostly we've worked in botanic gardens, and we assess the DNA content of these different individuals. Next slide. We've done this now for more than 15 species. I'll show results from a few of those species. They include magnolias, oaks, cycads, hibiscus, palms, and other species. Next slide. So similar to how you might think of animals conserved in zoos, in botanic gardens, we conserve plants and trees in meta collections, which were described earlier. Those are like plants distributed across many locations, many sites, many botanic gardens. And as Abby will talk about later, we can track these number of individuals across the whole botanic garden meta collection. So what's shown here is the number of plants for each of these species that are conserved across many botanic gardens and the amount of genetic diversity that's conserved in that number of plants. And I highlight 3 of the 11 species we've investigated have close to all of their genetic diversity conserved ex situ. The goal is to have all of it conserved. That's a hard goal, but what we're highlighting here is that many species do not have sufficient genetic diversity conserved ex situ. Next slide. Shown here is the number of plants in those gardens and the amount of genetic diversity conserved. And what you can see is that as we get more plants conserved ex situ, we get more genetic diversity conserved. Next slide. But it's not just about the number of plants conserved, it's also about how they're sampled from wild populations and how they're managed over time. So we have produced advice in collaboration with the Center for Plant Conservation and with BGCI. This document is one of the links that you'll see at some point in the chat. The meta collection document provides advice about sampling to better capture genetic diversity. So across multiple years, focusing on as many seed producing plants as possible, and sampling more when the species is highly selfing or low dispersal. This can help better conserve genetic diversity ex situ. Next slide. I'll talk about Quercus acerifolia as a case study. This, as Amy mentioned, is located in 4 populations in Arkansas, in 4 counties. It's quite well conserved in Botanic Gardens, and we have been working with the City of Columbia, Missouri, and U.S. Forest Service partners and others to establish a clonal repository, which will fully conserve the species' genetic diversity and also serve in the future as a way to produce seeds, acorns, for restoration, as well as, since it's in an urban area, to provide outreach to the public about this endangered species. Next slide. We have analyzed the genetic diversity ex situ and in situ. On the right, you can see the in situ populations. They each have a different color. On the left, you can see that one of those colors is not well represented. So that purple color, that population has not been protected very well ex situ. So we do need more samples from that population, according to our DNA analysis, to better protect it. We also find that in our ex situ collections, as you imagine, we have many half and full siblings. This is not great in terms of it can promote inbreeding and lose genetic diversity. And overall, we do still need more samples to provide duplication in case trees die due to disease or accidents or old age. Next slide. In addition to genetic analysis, we use GIS tools to help prioritize species. Which species do we focus on? Next slide. This method is basically a way to assess the coverage of a species' geographic range. How much have we covered it by our conservation efforts? Next slide. This is an example, Quercus georgiana, Georgia oak. You can see we can make a map like this and basically assess the white area, that's the total species range, and the black area is the area that we've covered with conservation collections. Next slide. This becomes a powerful tool when we do this across many species. So shown here are 4 additional Quercus species. Next slide. We can then summarize this kind of mapping exercise in simple charts like this. This shows a group of species that are well conserved on the right, and those species on the left need a lot more effort to fully conserve them. Next slide. We have done this not only with oaks, but we've also done it with pines, walnuts, hickories, beech, and a number of other tree species. And what we're finding is that small range species we've actually conserved fairly well. So those most threatened species we have conserved well, but we need a lot more effort on our larger range species, which can be threatened. Next slide. This helped highlight an oak species that is had very little genetic diversity conserved when I first started at the Arboretum. And we worked with the American Public Gardens Association and U.S. Forest Service Seed Collection Partnership, a great grant to better collect and conserve genetic diversity ex situ. This is Quercus havardii or Shinnery oak. And we found that it was previously only in 1 botanic garden, and we collected seeds across its range and sent them to 9 botanic gardens. And this method can also track improvement over time as we sample from more sites and get them into ex situ collections. Next slide. So we have found that genetic diversity in botanic gardens, as well as the literature shows, in urban populations, in street trees, genetic diversity is low. There's a lot of use of clonally propagated material. DNA studies can help assess the amount of genetic diversity and provide advice about improving it. And then tools like those GIS tools can help plan what species to focus on for future plantings and get more genetic diversity conserved in urban settings, and also raise public awareness about our threatened trees. Thanks very much.